नहीं है।
नहीं, मैं तो आई एम नॉट टचिंग QPM, so I just have one introductory slide, then you will explain.
आप बोल लोगे मैं बोल लूं, क्या फर्क पड़ता है, एक ही बात है।
नहीं, you are right. You are right.
रोमन को कह रहा था मैं एक का ही करवा लेता यार, दोनों में क्या जरूरी था।
नहीं, मैं भी यही सोच रहा था कि सर बता देंगे तो ठीक रहेगा।
मैं तो बल्कि हिसार आ गया, मेरे को तो बाद में पता चला ना।
Oh, I see.
आई एम एट हिसार, आई एम सिटिंग एट माय होम।
Oh, I see. सब कुछ ठीक है सर?
हाँ, घर से करना पड़ेगा। आप भी घर से कर रहे हो तो?
सॉरी?
यू आर आल्सो सिटिंग एट होम?
नो, आज ऑफिस है सर, वो सोइंग है तो फिर सिलेक्शन वगैरह करना था तो ऑफिस आ गया था।
चल भाई, मैं तो किसी काम से घर आया था। देखते हैं सबको बुलाया। 5 बज के, रोमन नहीं आया शुरू करने के लिए? क्या मैं कराऊं शुरू?
रोमन तो आ गया।
रोमन आ गया, रोमन शुरू करो भाई।
2-3 मिनट और ले ले क्या, क्योंकि मुझे लग रहा है कि मोर देन 80 से ऊपर पार्टिसिपेंट है सर, तो अभी 44 ही हुआ है सर। 2 मिनट सर, बस 2 मिनट के बाद कर लेते हैं।
इंट्रोड्यूस कराओगे तुम, 2 मिनट हो जाएंगे यार।
So, Dr. Raman, how are you?
I'm fine sir, fine sir.
Great. So, this is a 21 days training program.
Yes sir, yes sir. ये एसएल सर ने ये वो किए थे, क्योंकि जब इंडस्ट्री लोग से जाते थे सर, जब भी वो जाते थे कि एक ऐसा एक ओवरव्यू पूरा एक मेज का जो अभी जो हो रहा है सर, उसके लिए सर वो लोग ने सर रिक्वेस्ट किए सर से कि ऐसा कुछ एक ट्रेनिंग प्रोग्राम करे ताकि सारे ओवरव्यू मिल जाए करके सर, तो इसलिए रखा गया था।
So, the participants are from various organizations?
Yes sir, various organization, even from state department, line department also sir, extension worker, researcher sir, and sit sector से भी है सर कुछ, और इंडस्ट्री के भी है सर वो सर, तो इसे मोर ऑफ ए मिक्स है सर। हाँ
इधर कर दो, छोड़, हो गया।
फिरोज सर, अस्सलाम वालेकुम।
वालेकुम सर, कौन बोल रहे हैं?
सर मैं
बोल रहे
ओह, अच्छा, जहूर साहब। So, how are you sir?
मैं ठीक हूँ डॉक्टर साहब, आप कैसे हैं?
ऑलवेज ग्रेट जहूर। हाँ,
रमेश सर ने भी ज्वाइन किया है सर।
सब पूरा जहूर आज ये किसके नाम से ज्वाइन किया डॉक्टर जहूर?
सर बच्ची के नाम से ज्वाइन किया, ये उन्हीं का लैपटॉप में उसी का नाम है।
ओके।
वो हो जाता है या
नहीं, जरूरी है भाई, बच्चों का भी जरूरी है।
फिरोज सर, कुछ स्टूडेंट भी ज्वाइन करेंगे, आई हैव सर्कुलेटेड इट अमंग देम।
ओह ग्रेट।
रोमन, ये लीवर ट्रिम्ड क्या होता है भाई? दिस मीटिंग इज बीइंग लीवर ट्रिम्ड।
सर मैंने यूट्यूब में करके रखा है सर, उसको लाइव स्ट्रीम करके रखा है
सर। ये लाइव चलता है यूट्यूब पे भी।
अच्छा,
रोमन बड़ा एडवांस हो गया है।
लाइव स्ट्रीम ओके।
एडवांस, ताकि थोड़ा सा वो रह जाए कि कोई अगर सेशन बाद में भी देखना चाहे तो सर वो
अरे लेने दे, आनंद लेने दे। रोमन के राज में सारे काम है भाई।
बस नहीं सर,
रोमन तेरे राज
अभी शुरू करें सर? अभी
हाँ कर लो यार शुरू, कितने को हो गए? डॉक्टर साहब को भी बेचारा, ये देखो डॉक्टर फिरोज स्पेशल आपके काम के लिए ऑफिस बैठे हैं, इनको भी घर भी जाना है।
शुरू कर देते हैं सर तब से।
तो कर दो सर।
हाँ ठीक है सर। Good evening everyone. A very warm welcome to all the participants. joining us for this day 4 of our 21 days online evening training program. Today's program will feature 2 expert lectures under the broad themes of biofortified maize for nutritional security. And we are privileged to have 2 eminent experts which will share their insights on these important topics. And it is my pleasure to introduce our first distinguished speaker, Dr. Firoz Hussain, Principal Scientist, Division of Genetics, ICAR-IRI, New Delhi. Dr. Hussain is a renowned maize breeder and molecular geneticist whose pioneering works in quality protein maize, pro-vitamin A maize, and nutritional biofortification has made significant contributions to food and nutritional security. With more than 120 scientific publications and several prestigious honors, he is widely recognized as one of the India's leading experts in maize genetics and breeding. Today, he will deliver an expert talk on breeding for essential nutrient-rich maize status and prospects. Sir, on behalf of all the participants, I warmly invite Dr. Firoz Hussain to deliver his lecture. Over to you, sir.
Okay, so thank you very much. So at the outset, I also welcome all the participants, and then I would like to thank the organizers, especially Dr. SL, and then of course Dr. Raman, for inviting me to have interactions with you. Thanks to Dr. Ramesh as well, who is also an eminent expert in the field of biofortification of maize. I'm sure both the lectures you'll enjoy in terms of the product development and then how it is being popularized. So maybe I'll first try to share the presentation. Is my slides visible?
Firoz sir, visible है.
Okay.
Visible है, बिल्कुल.
Okay, so basically, I'll be presenting on work on essential nutrient-rich maize status and prospects. And as mentioned, I work as a maize breeder at IIMR New Delhi under Division of Genetics. And with me, we have Dr. Vignesh, Dr. Rajkumar, and Dr. Ashwin. They are now the scientists in our program. Along with that, we have our postdocs, PhD students, master's degree students, and of course, different project personnel. So the work which I'm going to present is basically a work of everyone mentioned over here. And of course, many of them are our past students who are now in different capacities in different institutes in the form of either faculties or as a scientist or a breeder, and even different seed companies. Of course, the importance of maize has already been taught. This is the 4th day. There is no need of re-emphasizing the importance of maize. It's a source of livelihood. And currently, we are producing 55 million tons of maize. And if you look into the utilization pattern, around 60% goes to poultry feed. Then around 20% goes to food. And with the current demand for ethanol, almost 18 to 20% are currently being utilized as a source towards of the maize grains towards the bioethanol industry. Now, if you want to start the program on biofortification of maize, you have to start with Quality Protein Maize, the pioneering work of Dr. Wassel, who was a breeder at CIMMYT, we all know, and then Dr. Villegas, she was the biochemist. And they basically developed with a set of cultivars, they call it as Quality Protein Maize, where the 2 essential amino acids, lysine and tryptophan, are almost present in the double the amount. Now, after me, Dr. Ramesh will present much details about Quality Protein Maize. So I'm not going to mention in details about Quality Protein Maize. Sir will present those things. So just quickly go through that whether any kind of breeding program we do in terms of quality aspect of it, molecular breeding is a key to its genetic improvements, because we normally follow genes rather than the traits, because the traits estimation is very costly. I have put a slide at the end. So molecular breeding is basically a very key component of this nutritional quality enhancement. And to start with molecular breeding, it was in 2008. One of our ICAR institute called VPCAS, they first came out with marker-assisted selection-based hybrid called Vivek QPM9, which is nothing but a QPM version of their field corn hybrid. So the normal hybrids called Vivek Hybrid 9. Both the parents were improved for the OPEC2 gene, and the hybrids were essentially then improved for lysine and tryptophan as well. And we at IRI also developed mass-based quality protein maize hybrids in 2017. And like that, there are several institutes under AICRP have now come out with excellent hybrids. Dr. Ramesh has a large number of QPM hybrids. Dr. Bhupinder has QPM hybrids from IIMR. Same is the case with our Srinagar Center as well as Vipika Center. So large number of biofortified hybrids for QPM have already been released in the system. But there is one unique thing that instead of OPEC2 gene, we at IRI have also worked on OPEC16 gene. There is another gene. OPEC2 is present on chromosome 7. OPEC16 is present on chromosome 8. When we combine both the genes, we found there is a almost 40 to 50% increase of lysine and tryptophan over O2 alone. So that is the greatest advantage. And unlike OPEC2, which causes the softness in the kernel, OPEC16 doesn't cause any softness in the kernel. So therefore, the kernels are hard, and it is not as soft as which many times we get along when we deal with or when we work with OPEC2 gene alone. So if you look into the cross section of the kernels, the deep orange color indicates the hard portion, while the white color indicates the soft portion. So now if you look into the photograph of E and F, the bottom two right side, central and the right side photograph, you will see the proportion of the orange kernel is very similar to the figure B. B is the normal maize, and C is the OPEC2 modified, that means QPM. So that's what it is, while A and D are the 100% soft kernels. So therefore, once the breeder starts working with OPEC16, the kernel becomes hard, and then it is a great boon to the breeder. Now we have several OPEC16 based hybrids are in the pipeline. There is another essential amino acid which is very important called methionine. Maize is poor in methionine, and the poultry industry, where 60% goes to those industries as a feed, they add a synthetic methionine. They also add synthetic lysine, but the synthetic lysine is cheaper while the synthetic methionine is costly. So the poultry industry will be greatly benefited when we develop a high methionine maize. In short, we call it as HHM, HMM. And another important aspect is currently there has been no methionine-rich maize released in the system. So we at IRI have started working, and then we added FLORI2 gene, which causes enhancement of almost 1.8-fold of methionine over its original version. Original was QPM plus ProA. So our improved version is having QPM, provitamin A, and methionine together. We also worked with another gene called aspartate kinase 2, and it also increases the methionine by 1.37-fold, little lesser than the FLORI2. And these ASK2 based hybrids are also done very well. So we are now trying to combine both the genes to study how both the genes can include methionine when they're together vis-a-vis when they're alone. We also observed very new information that there is a line called PMI PV5. It is one of our improved, one of our parental lines of released hybrids. When this line was improved for methionine, we found that there more roots are there, more root formations are there. So the number of seminal roots are more, root length is more, even the shoot is much vigorous than the original shoots. So the vigor index 1 and vigor index 2, these are the two parameters when we start doing the seed germination aspect. So it clearly indicates that high methionine maize will have a better field establishment in farmers' field because of the better root system. and therefore it will give a better establishment, and therefore finally the better yield. We are trying to validate those speculations, but this is a very interesting component of the study which we have done by improving methionine in our biofortified maize. Now, provitamin A is again a very important aspect. There are various colors of maize, light yellow, orange, deep orange, and all that. And normally we think deeper the color, higher is the methionine, higher is the provitamin A. But that's not the case. We found that around 3 to 4% are actually provitamin A. Rest all are non-provitamin A components like lutein and zeaxanthin. They only act as an antioxidant, but if you consume, they do not get converted into retinol, the chemical component of vitamin A inside the body. But then we got some CIMIC lines, and it was found that some of the lines had 80% of beta carotene, while the lutein and zeaxanthin is reduced drastically because it's a proportion. Now, two classical papers came in, one in Nature, another in Science in 2008 and 2010. And there are two critical genes. One is called CRTRB1, the red star section. So here what happens when beta carotene is formed, the mutant version doesn't allow the conversion of beta carotene into further components. So therefore, there is more accumulation of beta carotene. And there is another gene called LCYE, the green star component. So where the mutant version doesn't convert lycopene towards alpha branch or towards the lutein branch, rather the most of the lycopene goes towards the zeaxanthin branch. So thereby there is more synthesis of beta carotene. So these two genes we immediately introduced in our breeding program or in our germplasm, and in 2020 we came out with a hybrid called Pusa Vivek Hybrid 27 Improved. The original hybrid was from Vipikas. And if you can see, after almost 4 to 5 months of storage, while normal maize has 1 ppm of beta carotene, while our improved version has almost 5.5 ppm. And it's early maturing hybrids. So it matures in around 85 days, and 5 to 5.5 tons per hectare is the yield. So it is specifically for the hilly regions, not for the plain zones. Now in 2000, of course, before that we combined vitamin A and QPM together and came out with Pusa Vivek QPM 9 Improved. It happens to be the first double biofortified maize hybrids with QPM and provitamin A. It was again extremely maturing hybrids, and which is 1 ppm in normal maize, we have enhanced to almost 8 ppm, along with high lysine and tryptophan. Then in 2020, then we started improving the medium maturing hybrids. And then in 2020, we have Pusa HQPM 5 improved, Pusa HQPM 7 improved. They're all QPM plus provitamin A combinations, and yield potentials were very similar to their original version, which is otherwise was QPM. The original version is from CCSHAU Hisar University. We have improved here at IRI. Then in 2021, we have developed another set of hybrids, Pusa HQPM 1 improved, and then Pusa Biofortified Maize Hybrid 1, where both QPM and provitamin A were enhanced. So we follow two kind of breeding programs. One is we improved a very productive normal hybrids or the QPM hybrids into improved version. So it was a marker-assisted backcross breeding program. And simultaneously, we also used forward breeding approach, where we did we only genotyped the population for the foreground marker, and foreground marker means only selection for the target genes. Well, the background genome was new because it was a recombination between the two parents, and then we came out with several hybrids like Pusa Biofortified Maize Hybrid 2, then Pusa Biofortified Maize Hybrid 3. So they are not the version of any older versions, but rather they are the new set of hybrids with QPM plus ProAP combinations. Similarly, in 24-25 as well, we had several hybrid released through AICRP system for different zones. This is the map of our AICRP program, where you all know that maize, the whole country is divided into 5 agroclimatic zones. So some of the hybrids have been released for 2 zones, 3 zones, or even across the country. Now we had 2 hybrids, Pusa HQPM 1 improved and 5. So they were earlier released for Kharif season across the country. Now this has been now released for Rabi and spring season as well. Rabi spring essentially means except the hilly zones, all 4 zones comes under Rabi and spring. Spring essentially northwestern plain zone, and Rabi essentially the peninsular zone and central west zone and northeastern plain zones. We are also working on enrichment of vitamin E. Vitamin E is very important for cardiovascular function, neurological function. While vitamin A, which we worked earlier, we all know vitamin A is very important for vision, eye function, or various other components, body function as well. So we found there is a gene called VT4. VT4 converts gamma tocopherol into alpha tocopherol. Now, maize is very rich in gamma tocopherol. Gamma tocopherol is around 80%. Alpha tocopherol is roughly around 10%. But when we consume our receptor in the liver cell, they basically absorb alpha tocopherol more preferentially than gamma tocopherol. So therefore, if you can increase the alpha tocopherol, then the vitamin E activity is enhanced. So we could find there is a gene, there is an allele of VT4 which converts gamma tocopherol into alpha tocopherol very frequently. So therefore, or in a more number of conversion will be more per unit time. So therefore, we use this VT4 gene, and then we released, in fact, a triple biofortified maize hybrids, which is nothing but QPM plus Pro vitamin A plus vitamin E in 24 and 25. And they have been released for 3 zones and 4 zones. And they are also in the medium maturing group. And therefore, they had their yield potential, which is around 6 to 8 tons per hectare. Now, we have also improved sweet corn. We all know sweet corn is getting very, very popular. Now we have released QPM plus Pro-rich sweet corn. We call it as Pusa Super Sweet Corn 1, Pusa Super Sweet Corn 3, which has been released for 4 zones. So this, apart from the sweetness, it is also very rich in lysine, tryptophan, and provitamin A. And very recently, one of our hybrids, which is rich in provitamin A and vitamin E, have been identified. And this will be commercialized for commercial multiplication very, very soon. We have also enhanced the sweetness of the sweet corn by combining sugary 1 and shrunken 2 gene. So in Asia or in India, all our sweet corn hybrids are shrunken 2 based. But when we combine sugary 1 and shrunken 2, we could find there is shrunken 2 has around 25% sucrose, while when we combine both the genes, we have more than 40% sucrose. So the sweetness is more. So such hybrids are also ready for evaluation in the ICAR trial. And we have also developed sugary 1 based sweet corn. The purpose is not for commercializing for sweetness purpose, but for utilization of the specific grains, which is rich in phytoglycogen. Sugary 1 gene increases phytoglycogen, which is normally present in the grains by 5%. But now, because of the sugary 1 gene, it is enhanced to almost 25% or around 30%. So phytoglycogen is very important component for a cosmetic industry. And all the phytoglycogen which is being used in cosmetic industry are basically coming from traditional maize. But now the traditional maize has 5%, and now this type of sugary maize has around 25 to 30% phytoglycogen. So that could be used very well in this cosmetic industry. We are also working on high amylose. Normally maize has around 30% amylose, but now we have increased the amylose percentage almost 60 to 70%. There's a gene called amylose extender 1. And we have now developed several high amylose maize, which has also been found to be low GI. Normal maize has glycemic index of around 70 to 80, depending upon the kind of food we prepare or kind of bread we prepare. But the amylose extender based maize has a GI less than 50. So that is very good for the diabetic patient. And high amylose basically increases resistant starch. If you can see the pie diagram, normal maize has around 2 to 3% resistant starch, while our experimental hybrid has almost 20% resistant starch. So resistant starch means once we consume, it takes more time in the gut to get digested and absorbed in the body. So therefore, the increase in blood sugar sugar in the blood will be much slower as compared to the any kind of traditional food. We have also enhanced the oil content in the grain. The purpose is vitamin A and vitamin E are fat soluble. So if we can increase the oil, so therefore it will increase the bioavailability. So thereby the gain will be much more. So we have now enhanced the kernel oil by almost 35%. Normally it is 3 to 4% oil. Now our high oil maize has around 6 to 7% oil. And of course, the oleic acid has been increased by 50%, and palmitic acid, which is not desirable, is also reduced by almost 36%. So that is again another aspect of it. So the corn oil quality of these hybrids are much better because we have a better, higher amount of PUFA, that is polyunsaturated fatty acid, than the other types. The blue maize is also very important. So a maize rich in anthocyanin, in traditional maize language, we call it as blue maize because it gives a bluish tinge rather than a black color. So this anthocyanin is very rich in antioxidants. So now we have developed our hybrids, which is in the background of QPM, provitamin A, vitamin E plus anthocyanins. So again, a large number of genes are responsible for anthocyanin. So bringing all these genes together, it's a big challenge. And also there are several suppressors also there, which doesn't allow the formation of pigments. So that needs to be taken care of in the breeding program. We have also worked on increasing the bioavailability of iron and zinc. Normally 5% of the iron is absorbed in the human gut, and around 20% of the zinc is absorbed in the human gut. The rest goes through excreta and goes out of the body. So therefore, majority of the iron and zinc are not utilized in the body, and the culprit is phytic acid. Phytic acid is negatively charged. It's a salt. When it combines with positively charged iron like iron and zinc, so there are mutants called LPA, low phytic acid mutant 1 and 2. So we have used LPA 1 and 2, and then we developed 2 hybrids, PUSA Biofortified Maize Hybrid 7 and 8. They are low phytate plus QPM. Earlier from IIMR, they came out with the first low phytic acid based maize hybrids. And now we have combined the low phytic acid maize hybrids with QPM. And I'm sure these low phytic acid maize hybrids will play a very important role, especially in addressing the iron and zinc deficiency. We have also used all these biofortified maize grains for various food preparation purpose. So we collaborated with the Indian Institute of Hotel Management, which is an institute very near to within the PUSA campus. So they came out with cookies, biscuits, cakes, and brownies and other items. And of course, we tried with idli, dosa, and puri kinds of things, and they are very, very soft, tasty, and of course, apart from that, they are very nutritious. Now coming to this very important component, that why molecular breeding is required for breeding for these traits, which we have mentioned. Now, majority of the traits are to be analyzed through HPLC. For example, vitamin A, then vitamin E, anthocyanins, because not only we have to analyze its components as well. So we do have colorimetric method, but that gives the total anthocyanin, total vitamin A, or total carotenoids. We do not know the individual components. So it is very important in a breeding program to know the components as well. So HPLC analysis is the option, and per sample cost is around 2,000 to 2,500 rupees per sample. So if you want to convert a hybrid, which is made up of 2 parents, we calculated that roughly around 20 lakhs or 23 lakhs rupees are required if you want to breed for high provitamin A lines, where you are introducing a provitamin A genes from a donor to your recurrent parent. And every population which you generate has to be phenotyped for vitamin A through HPLC. But then we are using molecular breeding. So per sample cost or assay is only 30 to 40 rupees. So the entire thing can be done by around 1 or 1.5 lakh rupees. So you can understand how it's not only precise and how effective in terms of the cost management part of it. Now these are the benchmarks in AICRP trial. We all the breeders sit together during the workshop, and then we decide what are the baseline. The baseline level means what are the level which is normally available in the germplasm, and threshold means above which we'll call it as biofortified. And these are all very systematic studies done by several groups in our AICRP system. Once we come out with data, then we fix this threshold values. And because of this systematic approach in our AICRP, I think maize is the only AICRP in the entire country where we have so many traits for which threshold values have been fixed, and the breeders are working and and periodically new combinations of trait-based hybrids are being released. So these are the different combinations of traits which we are breeding, and these are their gene combinations. And so we initially started with single gene or single trait. Then we went ahead with 2 traits, 3 traits, 4 traits together. Gene-wise, they are 6 or 5. And then we regularly follow this molecular breeding approach for all the genes for which markers are available. The reference with star markers are the ones where we developed in our lab, while the other references are internationally available. So these are all public domain, whether it's been developed by us or by any international research labs. They are all in public domain, so anyone can use it. And if any one of you would like to start a program, please contact the ICAR-Indian Maize or us. We'll be able to help you. Now, this is the global map where country with the orange color have released vitamin A rich maize. And India developed first vitamin E rich maize in 2017, while China did it in 2019. But now, as you have seen, I think India is the only country where this much diverse types of biofortified maize are available. Globally, it is mostly QPM, vitamin A, and some cases it is zinc. But in India, we have vitamin A, vitamin E, QPM, then iron, zinc, then we have anthocyanins, high amylose. So there are various types. So I think we have a largest diverse type of biofortified maize available. A very important component is what happens to the yield if we increase the quality, because that is what is impression in everyone's mind, that whenever we increase the quality, the yield is reduced. So this is the ASRP data, where blue bar is the original hybrid and the orange bar is its improved version for one or the other traits, which is nothing but the quality traits. And the data suggests that there is no yield penalty. They are statistically at par, and that's how they have been removed. They all have been released in the ICRP system. And I'd like to make one of our a set of hybrids in Northwestern Plain Zone. In 2017, in the medium maturing groups, our hybrids had 64 quintals. And now in 2023, our biofortified hybrids had 84 quintals. We are telling the mean value across years in Northwestern Plain Zone. And our all experimental hybrids are now currently in 90 to 95 quintals per hectare. So there is a gradual increase in yield which we have been undertaking during the improvement of the breeding program. Same is the case with Peninsular Zone. Our first hybrid had 63 quintals, but now we have reached in 2026 when our hybrid is identified through ACRP, it is 90 quintals. So there is a sharp increase of yields in terms of in all these biofortified maize hybrids. Dr. Ramesh will also present similar data because that's what we'd like to convey, that now we are gradually mainstreaming the biofortification program with our field corn program. Our yield potential of all these biofortified maize in that maturity group, here mainly it is the medium maturity group, they're almost at par with, or not almost, in fact at par with field corn maize. So that's the reason why these hybrids are getting popular. We have also done bioavailability study of our provitamin A through National Institute of Nutrition, and it was found that it provides around 50 to 60% of the recommended dietary allowance in humans. Provitamin A rich maize also lowers the aflatoxin contamination, which is a boon for the Indian farmers, who basically stores these grains in a gunny bag, and because of the high moisture during the rainy season, it invites aflatoxin contamination through Aspergillus flavus. So that is one advantage. We have also studied the effect of QPM and provitamin A in chicken through Directorate of Poultry Research, and it was found that it has got a better body weight gain. It requires less feed, and the breast meat yield is more, while the abdominal fat is less. So that means this biofortified maize is more advantageous even as a poultry feed. Similarly, the low phytate maize was also analyzed, and it was found because the phytate was less, the iron, zinc, and phosphorus bioavailability was more, and the chicken had a better bone strength, and that is also one of the greatest advantage. And then because of the vitamin A, the color of the pigment basically accumulates in the leg part, beak of the chicks, and as well as the egg yolk. Now, the right side one where you see the 100% provitamin A, it has got a much deeper color than the control, which is otherwise yellow. So that clearly indicates that it has got a positive impact on the bird's growth and development. This is an international study, but I'd just like to mention that the grain color of the maize is inherently reflected in the yolk color. Especially for the non-vegetarians who take eggs as a part of the diet, for them it's very common that egg yolk makes a different color: white, orange, deep orange, light yellow, dark yellow, and all that. All these pigments are basically coming from the maize grains, and that's the reason why maize grains which are white in color is never used as a poultry feed, because white yolk colored egg is not preferred. So it has to be yellow, and it's yellow versions. Now it has been proven that when maize is vitamin A rich, so the vitamin A finally getting accumulated in the egg yolk is almost 10 times more than the normal yellow maize. So the poultry industry can get a benefit out of that, especially the layer industry, to a great extent. And of course, seed production is a very important component. Dr. Zahoor is here. He's a maize breeder from Srinagar. So we had an MOU, and then we shared all the parental lines of our biofortified hybrids. The left photograph is their farmers' visit to their field. And then in Kupwara district, we had seed production in farmers' field, which has been a great success. So Dr. Zahoor and his team through KVKs, they are promoting this hybrid, biofortified hybrids. Now, these are some of the last slides. Earlier, normally the field corn maize used to be very popular, and now still popular from the perspective of the seed industry. And now many of our biofortified hybrids have also been commercialized to different seed industries. And from 2022-23, we have started this commercialization. And currently this year, we had almost 75 MOUs have been signed, and almost 35 companies and FPOs have signed their MOUs, and 6 of our, basically 8 of our hybrids have been commercialized, and these are the values of commercialization. Normally, we give a license for 5 years where we share the parental lines, and they produce the parental lines, they make hybrids, they sell the hybrids, but the name of the hybrids cannot be changed. Whosoever is the hybrid from different institutes, the name has to be continued. So the increased commercialization over the years is an indication that biofortified maize is gradually improving, gradually getting popular. The main reason is it has now attained a similar yield as that of the field corn. So here we are getting the nutritional benefit as an additional component over and above yield. So that's the greatest advantage. And this is a photograph in farmers' field where we have licensed our hybrids to one of the seed companies, who in turn have grown it in Bijapur district in Karnataka in some farmer's field. And this is the photograph from that farmer's field. You see how beautiful the corn, the grain color is, and the plant type is. So that's how our all biofortified hybrids from across the ICAR system are getting very, very popular. Now, the major part is the takeaways. The first point is there is no yield penalty with any of these targeted nutritional traits, number one. Number two, many times people start thinking that there will be a trade-off when we start combining or stacking multiple traits. So as you have seen, we have developed multiple combination of traits, QPM, provitamin A, and vitamin E, but then there is no yield penalty. So there is no trade-off when we stack the multiple traits together. The next important point is deployment of the nutritional trait in the right genetic background is key to the success. The kind of background which you choose for introgressing those traits are very, very important. If it is high yielding, your version will also be high yielding. If it is low yielding, it will still be low yielding because the nutritional trait per se has no effect on enhancement of yield. And the fourth important point is, as I mentioned very categorically, the available biofortified hybrids are comparable with the traditional maize under same maturity groups. Now, there has been an increasing demand by the private seed companies for marketing of biofortified hybrids through licensing. And there is a significant impact of nutritional trait as a feed industry, as well as the food processing industry. So this is gradually being felt, and now they are coming to us for using these biofortified maize for various food and other purposes. But then there are challenges as well. So generation of awareness among public and the industry on health benefit is key. Providing premium or the minimum support price, especially for the biofortified maize, is something which will help further, although we are trying to mainstream it so that normal maize and biofortified maize are same as that of the yield potential is concerned. Then there is a need for establishment of linkages with food processing industry. Then there is always an apprehension that biofortified means low yield potential. So such kind of apprehension needs to be gradually taken care, because we have proven very categorically there is no yield penalty. Then, of course, now this biofortified maize needs to be climate resilient. We are now infusing such traits in our biofortified maize hybrids. Now, these biofortified maize grains can be included in various government-sponsored schemes like National Food Security Mission, then various other schemes. And of course, the production of quality seed is very, very important. So these are the challenges. I'm sure these challenges will be taken care very, very shortly. And now we are very happy to share that several of the biofortified maize hybrids across AICRP have been dedicated to the nation by our Honorable Prime Minister on 11th August. And Pusa biofortified maize hybrid 4 was one of them in 2024. It clearly indicates that the policy, from the policy point of view, the biofortified maize or the biofortified crop per se are getting a lot of attention from the policymakers, and I'm sure this will get more its due in terms of its nutritional advantage in coming years. So with this, I'd like to thank each and everyone. So this is a small team we are part of at IRI. They are all students, master degree students, PhD students, postdocs, and of course our scientists, project staffs are there. So I thank ICR, Director IRI. I would like to especially thank Director IIMR Ludhiana, entire AICRP centers, our partners, our funding agencies, CIMIT, Harvest Plus, USDA, for providing the donors. And of course, students and research scholars are our great strength. Whatever I have presented, basically each slide was a part of either MSc students' work or a PhD students' work. In a short time, it was really difficult to explain in much details, but I'm sure I could make at least the sense of it, that how biofortified maize hybrids have been developed, what are the traits, and how it is advantageous in terms of not only nutrition as well as it is at par with the yield potential, as having the similar yield potential as that of the field corn. So with this, I thank each and everyone for your patient hearing. I stop it here, and maybe over to each one of you. If you have any questions, queries, or anything, I'd be very happy to respond.
Thank you, Dr. Firoz, for putting light on in detail biofortified maize with different components. And perhaps Dr. Firoz has completed both lecture. Just I have to click in the next lecture.
The entire QPM part is with Ramesh sir, and I'm sure it will be another wonderful lecture.
Thank you, Dr. Firoz. You have
Thanks.
Delivered a wonderful lecture. And if any colleague or students, staff want to ask any question, please ask. Chandrasekhar here, very insightful presentation, timely research. Thank you.
So thanks to Dr. Chandrasekhar.
Sir, may I ask a question if I allowed?
Yeah, please.
Yeah.
Sir, this is Chandrasekhar from Bangalore.
Oh.
Sir, I am also working on corn, specialty corn. So it would be nice if you can throw some light on whenever we take genes like Shrunken2 and Sugary, how is the germination, sir? If there is any problem, and how you are coping up to address it, sir? Whenever because there is a food is very less to the embryo whenever it want to get germinated. So how to address that, sir? Can you just throw some light?
Okay, so it's a very pertinent question. So what happens is sweet corn maize, which is getting popular, is basically largely because of the Shrunken2 gene. And it is sweet because it inhibits the starch formation. That's why there is more sugar or more sucrose at the milk stage. But because of the lesser starch, the kernels are shrunken. So there is definitely a germination issue. So storage is definitely one of the very important aspects. But as a part of the program, or as a geneticist or as a plant breeder, sir, what happens is now there are various QTLs and modifier loci have been reported. So if you can bring in such loci in presence of the SHRNK2 gene, it increases germination. It has got a better vigor index. Vigor index means in a short time how the seed germinates with how much speed, how much volume. So that is basically a reflection of vigor index. Now, sugary one is having a more carbohydrate than SHRNK2. So sugary one has not much germination issue. SHRNK2 has germination issue. But then if you can select in a series of say 100, you have suppose 400 lines of SHRNK2, all are sweet, but then you will find that there are some lines which have got better germination than other SHRNK2. It is largely because of such loci being present in the background. So there are two ways: either if you have a best line as a combiner, but otherwise you SHRNK2 but less germination, so you have to bring in those loci into the background to have better germination. Or else, if you have an option to select among 500, 400 lines, the lines which is SHRNK2 but otherwise good in germination, phenotypic selection should be good enough. Now, coming to the second question, like when we combined SHRNK2 and sugary one, what happened? So it was definitely sweeter than SHRNK2. Its germination was affected definitely, but then we could select some lines which are as good as SHRNK2 because of again selection of modifiers. So such things are there, but overall, no SHRNK2 line can ever be as same as normal line in terms of its germination, vigor, or even storage for a longer time. And because of that reason only, the SHRNK2 seed is more costly. And that's why it is a premium aspect of it. So I hope I could answer your question.
Yes, sir, I agree with you, sir. The only concern is because very few farmers will grow with the help of drip irrigation, where they go for tray sowing and transplanting, where they can ensure 100% plant stand per acre basis or hectare basis.
Right.
But largely, if you go as from south to north, the farmers are using direct sowing. So this is going to be a very critical factor for the success of these kind of very rich seeds in terms of nutrition and all that. So that is where I was looking forward. Sir,
yes, you are right, but at the same time, I think this sweet corn cultivation is something where specialized farmers can or rather, if they take it up, then I think the benefit will be much more rather than it is cultivating as a normal maize. Yes, you are right.
Firoz sir, we did one thing with crop management in Shrunken background. We have been using a technique on at least 1 acre. I have done it successfully. We sow it in small polybags and transplant it. It's 100% crop stack, SH2 background.
Yeah, I mean, many of the farmers, they do it even for the field corn as well. But yes, at least it will help in germination. But large scale, what Dr. Chandrasekhar is telling, possibly large scale.
Up to 1 acre we have done, but sir, then it.
But if you have hectares of lands, I think that may not be a viable option. Yes, we have to look into that.
Dr. Chahur.
Sir.
At your place, temperature is also playing an important role.
Sir, उसमें वही है कि SH2 has less germination because when we sow it, it's cold.
Chandrasekhar, from which organization you are speaking? Kindly let me know your organization also.
Sir, I am actually I work for East West Seed.
East West Seed.
Yeah, I work for East West Seed, but this is just my personal interest. I have joined this.
Yeah.
Perfectly fine.
Yes,
yeah.
Perfectly fine.
Great.
And other colleagues also, kindly let us know the organization.
I did visit IIRI. I met Dr. and all people.
Oh, very nice.
Yeah.
Firoz sir, there's one more question in the chat. Prahlad or some, I wrote a little bit, but I'll refer it to you.
क्या है भाई कौन पूछ रहा है प्रहलाद development of nutritional enhanced maize phenotypes with combined quality should focus more from initial stage of development. It's generally.
No, such thing is already there. It's QPM, provitamin A, and zinc indirectly through low phytic acid is already there in our system. Vipicas has developed. In fact, we have also developed. So yes, such products are there in the program itself. Yeah, so there's another question, degradation of provitamin A. Yes, we have published an article and it shows that provitamin A degrades very fast during the first 3 months. And but at any point of time, the provitamin A rich maize grains will have much higher provitamin A, even though degraded, over the traditional maize. That is the advantage. So, for example, at harvest, suppose the provitamin A rich maize has 10 ppm. Or say 15 ppm, and normal maize has 5 ppm. So there is a 10 ppm difference. After 6 months, the provitamin A rich maize will have say 7 ppm, while your normal maize will have only 0.5 ppm. So it is reduced, but at any point of time, it is much higher. Number one. Number two, there is another gene has been identified called CCD1. So that is responsible for degradation. So now we are trying to combine CCD1 allele into our provitamin A background. So during storage itself, it is the degradation is reduced to a great degree. So that part is also being taken care of. But at any point of time, as I mentioned, it is much more advantageous than normal maize.
Anyone else? If no more question, then on behalf of Dr. Roman, Dr. Sankarlal, both are perhaps not. Roman is there. Roman?
Yes, sir. Yes, sir.
Kindly read out your formal vote of thanks to Dr. Firoz.
Thank you so much, sir, for an excellent presentation, sir. And so you have covered of many aspects of the this one nutrition nutrition QPM vitamin aging related all the issues and all the major status and prospect has been covered, sir. Thank you so much for your nice presentation, and we have a nice interaction also, sir. And for the second presentation, sir, we will move.
So 1 minute, Dr. Raman. So I once again thank the organizers, Dr. SL, you, and of course Dr. Ramesh is here, and all the participants for their patient hearing. So anytime any help required, please may be contacted. Dr. Raman may be contacted. So thank you very much.
Sure, sir. Sure, sir. Thank you so much, sir.
Thank you, Dr. Firoz.
So moving forward to the next presentation, sir, our next speaker is Dr. Ramesh Kumar, Principal Scientist at ICAR-IIMR Ludhiana. Ramesh sir is a leading maize breeder. His works mainly focus on QPM maize, and he has developed many QPM varieties as lead developer. And his work on QPM has made significant contribution. And as I have told you, many varieties has been released as a lead developer. Today, sir will deliver a talk on quality protein maize for nutrition security. Sir, I invite you to deliver the lecture, sir. Over to you, sir.
Okay, Dr. Roman. Just I'm sharing my slides. It's okay now. It's okay.
आ गया सर.
आ गया. पूरी तरह से नहीं आया, पूरी तरह से आया.
नहीं, आपको उस पे डालना है. हाँ सर, perfect.
Okay, Dr. Roman, thank you very much. And Dr. Firoz has already delivered a lecture in detail about all the aspects. We are covering the quality aspects, including this quality protein maize, provitamin A content, and other nutrient also. So I will deliver a small lecture. Rather than doing all the things, I wish to skip the slides which Dr. Firoz has already presented, and I have taken some slides from Dr. Firoz also. So this is the malnutrition global status, which just a minute. More than 2 billion people, they're suffering from micronutrients, and Dr. Firoz has provided this slide, and approximately 8 million people, they are undernourished. Major problem in case of less than 5 years age of children. Under 50 million, they are stunted. Then approximately 50 millions are wasted, and approximately 40 millions they are overweighted. And malnutrition contributes to loss in Asia and Africa majorly, 11% of their GDP in both these Asia and Africa. And in details, you can go through. These articles are published, and check out this website also, UNICEF, WHO, World Bank also. You can check this. This is a bigger issue. So how we can solve, we should think our country is planning in different way, and worldwide also policies are making to solve these issues. In India also, this mapping showed that 12 to 16% in district, and this stunting is out of 640 district to 239 district. They are suffering for stunting level that more than 40%. So in our country, you can see this approximately 50% population is facing these stunting issues. The growth is not normal due to malnutrition issues. So these are the some government has put surveys. In first survey, on different aspects, neonatal mortality rate, infant mortality rate, etc. The situation was alarming. And in another survey, which was conducted after a few years, few aspects they have improved. And the green bar, this neonatal mortality rate per 1,000 births, it was improved. Likewise, in child less than 5 years mortality rate, it was also improved. But in case of overweight or women's having overweight, men's obese or anemia, anemic conditions, they are still alarming rather than they are increasing. So we have to think about this, and the government has put more emphasis, more money is to be pumped for solving these issues. These all are related to malnutrition. So out of these 17 goals, you can see this major 12 goals belongs to this malnutrition issues. Government has fixed some sustainable development goals, and from out of these SDGs, you can see the malnutrition issue is again highlighted by the government. And core issue is to eliminate extreme poverty, hunger, and malnutrition. So we are emphasizing gender equality, good health, zero hunger, no poverty. All these are related to these aspects. Then in GDP also, in the first slide, I have showed that Asia and Africa, they are more, as far as GDP is concerned, 11% GDP is wasted, or we have to invest more to solve these issues. So when we are putting money for solving the malnutrition issue, if we are in normal terms, or you can say, we can say that when we are spending 1 rupee, we are getting benefit of more than 15 rupees. We are in terms of dollar, we are spending $1, we are getting benefit of $15. Because when your population is more healthy, they can work more, the country will get more benefits. So at our institute, what we are doing, Quality Protein Maize, we are working to improve for high lysine and in addition to this, we are also started working on high methionine maize also, provitamin and low phytate vexicol. And though Dr. Firoz has elaborated each and everything in his presentation, so I will put some light on different aspects. What is biofortification? How we are developing the genotype with improved one, either through conventional breeding or biotechnology, without sacrificing any agronomic performance. We are improving the maize with a particular quality trait. For example, we are talking about quality protein maize, the lysine and tryptophan for which we are working. The lysine and tryptophan in case of quality protein maize, you all know that are approximately doubled. The tryptophan is more than 55% than the normal maize. And biological value again, in case of normal maize, it was below 50%, and in case of quality protein maize, it is increased 80 to 90%. Intake utilization also, it is doubled, and the maize requirement is very less in case of quality protein maize. So the quantity is reducing, and quality is increasing, we need less grain to improve our health, to provide the required level of lysine. We are getting more lysine tryptophan from the less quantity of maize. So this is very good that high biological value is there. So less intake, more benefit. Then we are talking adult requirements also according to WHO or in Institute of Medicines also. In both cases, you can see the adult requirement in case of non-QPM. It was 46 and intake to meat requirement was 490 grams per day. Where in case of this quality protein maize, again, the requirement is less. So in both the cases, the less quantity is required to meet out the requirements for adults. And so how we are developing this quality protein maize? This is a very old slide. And as Dr. Firoz has already he has shown that quality protein maize, which was developed by Dr. Vashal and Velagas in 1964, March 1964 also, they have demonstrated what is the OPEC2 and how we are developing OPEC2. You can see when you are putting OPEC2 seeds on the light will not pass. 100% OPEC is there. What we need, but that 100% OPEC kernel that are soft, chalky kernel. And again, there was an issue of storage. Stored grain pests were more. So to avoid that, to solve that issues, again, the breeders started working to have hard kernels, vitreous kernels. So then with the help of light screening board, we used to screen all the grains. And from starting, we used to start 75% opaqueness or 50% opaqueness. Then slowly, we are selecting the grains with 25% or around 25% opaqueness is there with threshold level of lysine tryptophan. So by using light screening board, we have developed so many lines, and by using those lines, we have developed so many hybrids also that hybrids are giving higher yield along with the required quantity of this lysine tryptophan. And that yield level of these hybrids, which we have released recently, is touching 10 ton or more than 10 ton also. And the hybrids which are in pipeline, they are giving as good as in case of field corn. One of my colleagues was asking in case of sweet corn. So when you grow quality protein maize, improved quality maize right now, nowadays, you will find same yield level as good as the field corn. And Dr. Firoz has also showed one slide that how they have quantified the losses in yield and how we are mitigating that. So this is the impact of quality protein on human being. The low quality protein as compared to milk. As you know that milk is too costly nowadays, and most important is purity, availability, and perhaps as per record or reports, more than 65% milk is spurious. So there is also hazardous to your health. When you are getting quality protein at a cheaper rate and with required quantity, you can, generally we used to say that 1 glass of milk and 1 chapati of quality protein maize, that is equivalent. Because the casein milk bioavailability is 90%, and bioavailability in case of quality protein maize, it is more than 80%. So it is as good as milk. It is meeting the energy and protein needs of the infants and children rather than adult person. It is very good for infants and children also, improving growth rates in young children. And all these, this is solving different health issues. So in case of poultry, as well as in case of pigs also, piglets grew 3.5 times faster on OPEC2 or quality protein maize as compared to the field corn or normal maize. In case of QPM, 3.5 times more growth as compared to the normal one. So you can see this pig fed with the quality protein maize, the bigger one, and the smaller one, this is the fed with field corn maize. So this is a old slide and published by CIMIT. So the requirement, average requirement per day, 21 gram only. Whereas the pig fed with QPM based diet gained 256 gram per day. Weight gain, as in case of normal maize, it was only 21 gram. You can see the growth level by feeding this animal nutrition feed made up of this quality maize. So there are various benefits. It is providing food security, high biological value, improved dietary supplement. All these I have already presented in earlier slides. This is also I have already covered, and this slide, first time when Honorable Prime Minister has dedicated biofortified crop varieties to our country on the World Food Day. At that time, these three quality protein maize hybrids were also dedicated by the Honorable Prime Minister, and this belongs to our institute. This is pride not for us, but whole ICAR-P also. So, as Dr. Firoz has already shown some slides that they are working on multi-trait quality lines. Multi-trait, they have released so many hybrids. So you can see this slide in red mark. I have already mentioned that in case of lysine, tryptophan, protein content, provitamin A, and phytic acid also, we are also working all these aspects, and we are developing germplasm with high lysine, tryptophan content, high provitamin A content, along with low phytate also. So we, at our institute also, we are putting emphasis that we must develop such lines and such germplasm and provide to ICAR-P system so that our partner develop more and more hybrids, quality hybrids with multiple traits, multiple quality traits. And this methionine content also, Dr. Firoz has developed hybrids and has shown slides that we have developed these lines and then developed hybrid, and they have released also. So at National Center as a nodal agency, we are responsible for developing germplasm, and we have developed such germplasm which is rich in methionine content along with lysine and tryptophan. So though they are negatively correlated, but we are successfully selected lines, and these lines have been registered at NBPGR also. Recently, we got registered 5 lines, and these are all available for national system. They can take advantage of these lines, and they can include the breeding program. And in future, we are also giving, would like to give methane-rich hybrids also. So to improve the lines, to develop new lines, we are developing lines by using DS technology, and we are developing lines through conventional breeding. We are developing lines through marker-assisted selection, backcross breeding. It's Dr. Firoz has put light that how they are putting the particular gene in their elite line background. So we have developed a few lines by using this DS technology, and this is the per se performance at Ludhiana location. Ludhiana location is not good for such purposes, even though because seed production is majorly based in South India or Eastern India, and if you want to say the per se performance of the lines, because the female lines is productive, then only hybrid will be successful. So we are selecting the lines which are giving higher yield inbred lines, so that we can have the seed production ability. More seed may be produced by using such lines when they are female parent of any line. So when we use these lines, DS lines, and crossed with our testers to see the test performance, you can see this at Ludhiana location, even few of the hybrids, they have given more than 10 ton yield. And at Mandya location also, most of the hybrids, they have given around 10 ton yield. So yield level in combination, these lines are giving very good yield. And this is new germplasm which we have developed, and you can see this Bio 9544. In both the cases, this was a field corn check we have put in a trial of having all entries from the quality protein maize group, and you can see the performance as compared to the field corn. Performance of our newly developed hybrids is as good as field corn. This is we have developed few lines by using different combinations with different germplasm by using backcross method, as good as is marker assisted, but field corn breeding also, and we have developed very good lines which are giving per se perform very good yield in these cases also. And again, we have developed hybrids by using these lines by crossing with our testers, and you can see at Karnal location, because Ludhiana location we are having sandy soil at our farm, and Karnal we are having good soil, so the yield performance there is a difference. Even though this is a low yielding environment at Ludhiana due to poor soil quality, the yield level of our hybrids is approximately 90 quintal or 9 ton. Whereas in case of this field corn hybrid, they are also giving at par yield. And at Karnal location, number of hybrids which we have newly developed by using these lines, they are giving more than 10, 12, 13 ton yield level. So we have developed our material to different authority group also, and now I will come to our real work, which Dr. Roman has already told that we have developed so many hybrids, and these hybrids are available in market, or you can say a few companies has already signed MOU, and many more can sign MOU. You can see the grain color as well as the yield level, though it is giving average yield was 72 quintal or 7 ton in our AICRP trials, but it is having more potential. It is having approximately 10 ton potential, and with required level of lysine, tryptophan, and disease insect also. You can see this data. We have already released these hybrids, and this is the IQMS 203. This is the rain-fed ecology hybrid, and many companies have signed MOU for these hybrids with our institute, and they are particularly providing seeds in Rajasthan, Madhya Pradesh, and a few years back, 1,000 quintal seed was produced by the company, and they supplied all to farmer field through state governments. And right now also, there was a tender having requirement of these hybrids, 20,000 quintal hybrid seed, and a few companies they have already signed MOU, and they have produced seeds also. I am in regular touch with this, and they are asking me so many questions, and they are putting photographs through WhatsApp for quality measures also. So this is a very good hybrid, and this is a national check. And I am happy to share that Dr. Firoz is there, Dr. Zahoor is there, and many more colleagues are there from AICRP, that this is the must hybrid which you can see in AICRP trials, and giving more than 10 ton yield in the AICRP trial. Because
sir, unbeatable check.
Yeah. So this is very good hybrid, because in AICRP they all are irrigated under irrigated condition. Zahoor is witness to this hybrid. This is giving more than 10 ton yield. These are the few new hybrids, 206, 207, 208, and recently notified in 2025. 206 is for zone 3 under in northeastern plain zone, Bihar, Jharkhand, Odisha. This is a high rainfall in Kharif season, and due to erratic rainfall, yield level of any hybrid is not so good, even though this has given more than 6 ton yield. And when we are testing this one company, they have taken sample of these hybrids from me and grown in Assam. He called me that this hybrid is beating all field corn also, and that Dr. Zahoor has also grown at his place. So this hybrid is very good hybrid, giving more than 10 ton yield in under such conditions. And
sir, all of them are 10 plus, all of them.
Yes, sir. All are 10 plus. 207 again recommended for zone 2 and zone 3. Both these environment, you can see the difference of zone 3 and zone 2. In zone 2, the irrigated environment, the yield is more than 8 ton in AICRP trial, average yield. And you can see this again, 208. This is a rain-fed ecology, 7 ton yield under rain-fed ecology. And these are the 209. This is newly released for zone 1 for Zahoor, Dr. Zahoor's area, Assam, Mizoram, Arunachal Pradesh.
Yes.
This area, this is again a very good hybrid. And
sir,
you have
is it 209? Now it is here, yes.
Yeah, 209. We have recently notified it. And this hybrid is having higher yield than our already released LQMH1 in all these states. This 210, this is again a high yielding hybrid for zone 2, northwestern plain zone, with all including quality traits and having resistant to this chylopatulus as well as the MLB, major disease, VLSB also. So this 211, this is again for 2 zone, northeastern plain zone and central western zone. Dr. Zahoor, today morning I have received a phone call from one private partner. He wished to sign MOU for whole India. He asked me to provide hybrids. Then I requested him to visit IIMR, and I will provide hybrid for each and every zone, each and every corner of the country.
That will be wonderful, sir. That will be wonderful.
Yeah. 212, this is again that is for this zone 3, and 213, 214. 213 is released for central western zone, again rainfed ecology, and 214. This is the first hybrid released for zone 4, peninsular zone. Now my colleague who was from perhaps Bangalore or somewhere else, he was talking about sweet corn and asking question from Dr. Zahoor. Sir, this is an excellent hybrid giving more than 10 ton yield. This is average yield, you can say 9.4 ton in AICRP trials. A hybrid which is giving 9.4 or 9.5 ton in AICRP trial, and at larger scale under management, good management condition, it is giving more than 12 or 13 ton yield. There is slow growing, late, medium to late maturing hybrid, excellent hybrid, and in rainfed ecology also it is giving 8 ton yield. So this is an excellent hybrid. And in addition to these slides, Dr. Firoz has already shown to all of you that our colleague, they are working on different aspects, USA HM8, then HM9, and the QPM plus provitamin A. This was presented by Dr. Firoz, and this HQPM 7 improved, HQPM 5 improved, all these our colleague, and this VL QPM hybrid 59 by our Almora Center recently released, and Dr. Zahoor has also released this one hybrid for Jammu and Kashmir state. This is also a good hybrid. And again, Dr. Firoz has put light that how they are working on reducing the phytate in maize, and the higher phytate content that is affects the bioavailability of iron, zinc, potassium, magnesium, and calcium, all these vitamins. And we are working on this aspect. We are developing germplasm for low phytic acid, and we have converted one hybrid by using this. This is the technique Dr. Firoz has also already explained. And what we have done, that PMH1, PMH6, these 3 hybrids we have taken, and we have worked on this and developed. This is the first PMH1 LP low phytate version of PMH1 we have released, and you can see this level of this phytic acid also we have reduced 1.89 milligram per gram in case of PMH1 LP. This is the level of low phytate, whereas the threshold level is 2.5. So we have reduced it to very low level, and this hybrid is very good, improved hybrid, having high yield for northwestern plain zone. Then we are also working on high amylose also, these different groups. We have categorized our material and then try to develop new germplasm by using this magic pool. We have developed these different pools by taking the genotypes from the different clusters, and we have developed magic pool 1. Likewise, magic pool 2 also we have developed. And then you can see this driving new germplasm lines. The genetic base for this Vexi is, we have very narrow genetic basis there. So we wish to diversify the germplasm base for Vexi maize. We have taken few private hybrid also, and this is the Vexi genotype which we have already converted And by using this UMI 1200 Vexi, 1230 to 1201, we have already converted these lines into Vexi version. So these private hybrids also, again, we are using to develop huge germplasm and atotic pool 1, 2. We are categorizing our material, already categorized material. You can see the amylopectin percentage in case of atotic pool 1 and 2. The range is 84 to 98%. So up to 98%, we have increased the amylopectin in case of this pool 1 and 99% in case of pool 2 also. And we are making new cross combinations by using these two pools and developing hybrids. And we are also studying the yield penalty in case of Vexi maize. People used to say that Vexi maize is giving less yield, or approximately 20 to 25%. Yield is low in case of Vexi hybrids as compared to the field corn. And we are trying to select the germplasm which is having less reduction in case of maize. So we are conducting such trials also. Dr. Firoz has shown that journey, that how the yield level is increased in case of this biofortified maize hybrids from 6 ton to we have already released 10 ton or 12 ton hybrids. In all these cases, I have not put that slide in my presentation. So though I have given to someone else, today I'm out of my station. That was my office computer. It was not available with me right now. So yield level of the quality protein maize or biofortified maize, we have increased from 6 to 12 ton, approximately double right now, and these hybrids are available. And in case of quality protein maize also, we are developing germplasm for this high amylopectin content, or you can see the Vexi maize. So we have initiated this program, and these lines are right now available. We have completed foreground background selection, and you can see this 2192 and 106. They are parents of this IQMH 203, and this is background recovery. And you can see approximately 90% background recovery we have already covered. And now in the foreground selection, we have selected the lines with targeted gene. And after recovery of the background, now we are in this stage. The lines are finished lines, converted lines, and we have made crosses, and we have already entered these hybrids in QPM based this high amylopectin hybrid, or you can say Vexi, QPM Vexi in AICRP. And in coming years, you will be able to see this again, this national check with the improved version. So in our molecular breeding program at our institute, though I have already explained that we are putting emphasis in high amylopectin, lysine, tryptophan, provitamin A, low phytate. We have started working on different diseases also. Right now, we are targeting sorghum downy mildew. My colleague Dr. Chikappa is working on this, and he has initiated this program. In coming time, or in coming future, you will be able to see this SDM resistant total hybrids. Particularly, this issue is in Karnataka, and we will be able to solve this issue. And this is the Vexi maize hybrids. One slide I've shown for quality protein maize. This is the field corn hybrid 282-92. These two already released hybrid we have taken and we have converted these hybrids. And again, you can see this range of recurrent parent genome. We have recovered this approximately 90% in all the cases. And this is the yield level you can see. And I'm happy to share that one hybrid we have released during this workshop. This will be notified very soon. These are the converted version. You can see, again, this is the level of nils, or our newly released hybrids by using nils also. The germplasm which we have developed under this molecular breeding program, we have used these lines in our conventional breeding program also, and we have developed and released hybrids, different hybrids. So at our institute, we have conducted few experiments for sensories, or you can see this how the quality is affecting by cooking, or how we can use this. So we have made chapatis from quality protein maize as well as field corn. You can see there is no cracks in quality protein maize chapati, and in case of this field corn, there are some cracks, and the chapatis not so good quality. The quality protein maize is taken less water for dough making, means it is very easy to make dough and easy to make chapati as compared to the field corn. And when we see the shear force required to break down that chapati, in case of, we have taken 7 genotypes, this quality protein maize 1708 means 203. And in these two quality protein maize, you can see the very less shear force. Where less force was required to break down that chapati, these were as good as landrace. In case of landrace also, very less force was required to break down that chapati as compared to the field corn. White maize hybrid or yellow maize hybrid, you can see the force was required from 4.77 to 5.98 or 6.96. So very high force was required to break down the chapati which was made from this field corn. And in case of quality protein maize, it was as good as landrace. People used to say that landrace or local desi makki is very good for this food purpose. So you can see the performance, you can see this data, this chapati is so soft as good as in case of the desi makki. When we compare the flour or chapati, people used to say that after cooking, different quality traits, they are getting reduced. But the lysine and tryptophan, when we estimated from flour or chapati, because in grain, it was approximately 1% tryptophan level. In case of flour, when we crush these grains to make flour, temperature in chakki, you know how much temperature is there. And again, when we are making chapati and baked, the chapatis was put on tawa and gas heat was there. Even then, the quality, the lysine and tryptophan, both were intact in case of chapati. So no reduction at all. When you are taking quality protein with chapati, means you are taking a quality food. So these are based on sensory, different sensory, color, aroma, taste, and overall acceptability. Quality protein maize was champion. These survey sensory attributes were estimated while putting all these samples with a code to many people, and then asked to give preference as per their liking for all these color, aroma, taste. Then we averaged all this data, and this is the table of that data. You can see this overall acceptability for quality protein maize. So quality protein maize is excellent food for humans, excellent food for infants, children. You can make so many products. You can serve to anyone. You can serve to your guest. And these are the value-added products which we have made from quality protein maize, QPM burfi, QPM cookies, and biscuits. All these we are making at our institute. And we have one small unit of agribusiness incubation. Any farmer, any student, any small enterprises, MSME company, if they wish to take any training, they can write a mail to our director. We will make arrangement free of cost for 1 day or 2 day training. And Dr. Roman is coordinating all these activities as an extension worker. I'm highly thankful to him. We have given such trainings to hundreds of people. Many times we have organized trainings even at by reaching out at Begusarai, Patna, or any far-flung place. The farmers or ladies farmers, they cannot come. We are going there to provide such trainings. So you can see this all these infant foods, porridge, chilla, sattu, biscuit, kheer, halwa, upma, everything is made up from this quality protein maize. You can see this preschool children food also. All these puri, katta, khamman, crispy. In South India also, in North India also, in Eastern India also, there are so many products which at different institutions they are making from quality protein maize and providing to the required people. Other products, chapati I have already explained, and papad, etc. Number of products, and we have published folder, we have published one more booklet, technical bulletin also on value-added products. You can visit our website, and if you got a chance to visit our institute, you can see that you can take out one handout or one publication from our institute also. And one more important thing, corn silage. This is very important and very cheap source of fodder. Green fodder is always required for milking animal, and nowadays this silage is made up of along with grain. They are farmer, they are saving the money they have to spend on this dry grain. What I forget the name of this product. So they are putting grains to save money on from that also. And quality protein maize, because it is rich in different aspects. Biofortified maize, it is having high lysine, high tryptophan, along with all other traits which Dr. Firoz has already explained. So we can use that maize for silage also, so that our milking animal, they will get the biofortified fodder also, biofortified silage also. So this is a very good business. Anyone can start. In Punjab, perhaps more than 50 units are there. They are operating a farmer field. They are taking 1 acre land and they have put this silage plant, and they're nearby. They are purchasing crop from the farmer, and the farmer is easily getting 40 to 50,000 in 3 months crop, and without wasting money on harvesting, without wasting any money on threshing and other things. So he's getting 75, 80 days, 89, 80, maximum 85 days crop. He's selling standing crop. Farmer is very happy, and some farmer, they are taking 2 crops in 1 season. Kharif and the late Kharif, he is enjoying, and the people, they are getting this milking animal, particularly dairy area. I am sitting at Ludhiana, and when you got a chance to visit the Mandi, this dairy area in morning, there are dharam kanta, the automatic weighing machine are there. So you will see this one tractor trolley will come, and people will be there in queue. They will purchase this silage. Silage, you can see this one ton weight packet, and they will go to their home, go to their dairy, and then they are using directly. No need to store. They are purchasing on daily basis, or they can store it at their place, and they are feeding to their animal. Particularly in May, June, July, there is no green fodder. At that time, they are providing a better food to their animal, and the milk yield is not reducing during the particularly the dry period. And there was a parliamentary standing committee visit last month at Ludhiana. 19 MPs, member parliament, were there, and our nearby university, Guru Angad Dev Veterinary University, they presented very well that the silage is working, wonderful working in Punjab. Not only Punjab, they are exporting to different states in our country, and they are providing the better feed and food and improving the milk yield. Thank you very much. This was in so brief. Now I'm stopping share my slides. If anyone wishes to ask any question or interacting with me.
Thank you so much, sir.
Thank you, Dr. Roman.
जहूर सर, अगर है तो
नहीं, let the audience ask, क्योंकि हम तो रमेश सर और मैं तो almost रोज
हम तो colleague हैं, we are interacting daily.
अगर किसी का अगर सवाल है तो
let the participants please ask questions. You are the best of the expert here, please.
Yes, sir. किसी का सवाल है तो बताइए.
Perhaps Dr. Firoz has covered all these things.
Yes, sir.
None is.
नहीं, अगर किसी को ऐसे डायरेक्ट ना पूछे तो लिख भी सकते हैं चैट में.
Yeah.
Sir will answer.
They can send to Roman also if they need.
Yes, sir.
further clarification later on also we can give.
Yes, sir.
Sir, I have a question.
Yeah.
Yes, please.
Yes,
Yes, please.
Sir, the very interesting insight, sir. First of all, thank you for that. And in terms of fodder, sir, fodder and silage, present day, there is a high demand on fodder.
Yes.
Especially urban areas where some people follow this dairy and all these. So they need this green fodder. Are there any studies, sir, in terms of, corn, specialty corn comparison? And there is an increase in the milk yield in the cows or buffaloes. Are there any such
Dr. Chandrasekhar, right now I don't have that slide, and I already said that Jagadwasu, Guru Angad Dev Veterinary University, they have shown one slide. They have explained very well during Member Parliament visit. There was a big meeting, approximately 19 MPs and parliamentary general secretaries and secretaries were there, along with the staff from the university and the institutes. They have explained at length that this is the feed or fodder which we are providing through silage. That is increasing yield because the taste is very good. When you open this silage packet, achar-like smell will be there, and animal will run out for that. It is very tasty. So milk yield, this quality of this fodder, which we have tested, our biochemist has tested that green fodder as well as the silage, there is no difference in quality. So as good as green fodder, we are providing to milking animal. All of you know that milking animal, they need green fodder in their diet. And when the dry period is there, more heat is there, very less green fodder is there. At that time, automatically the yield level is decreased. But when we are having silage, so during particularly dry period, there will be no reduction in milk yield. That is beneficial for us. And I have seen at Ludhiana, because I used to come for morning walk sometimes and for exercise. I have seen so many tractors and they're full of with this silage and they're selling during dairy. There is one point, Ramkanta. At the Ramkanta, people used to come with their vehicle, they're taking it. So I don't have slide, but in future, if you need that, I can take that slide. And
so we have another session on this silage aspects on 4th of July.
Manish, very good.
Yes,
another session is there, then you will having more
The
insights for silage.
Pradeep sir will be taking the session, sir, and I think he will clarify more on
then
this.
I will. Then I
some
will
of
ask
from
Pradeep also.
silage industry also, sir.
Oh, silage industry also. Then it's good.
Yeah.
Then it's good. Then there will be no issue. You will
Thank you, sir. Thank you.
Thank you, Dr. Chandrasekhar.
So, if there is no further question, then I'd like to thank Ramesh sir for a wonderful presentation, sir. And thank you so much. And with that,
Mr. Tiwari, Dr. Tiwari is asking that in Bihar, any option to get training on offline mode? Yes, we have given 2-3 trainings in Bihar offline. We visited our institute at Begusarai along with our staff, and we have provided training. In future also,
वहाँ पे अगर किसी का अगर ट्रेनिंग अगर करना है तो मेरा हम लोगों को कांटेक्ट करिए हमारे डायरेक्टर सर को. वहाँ पे हमारा शायद लास्ट ईयर हमारा करीबन 5 से 7 ट्रेनिंग हुआ है और आत्मा के थ्रू हुआ है. तो वो हम लोग हर 2 महीने में ट्रेनिंग करते हैं बिहार में भी
करते हैं.
तो हाँ, तो आप
हमारे इंस्टीट्यूट का सेंटर है. वी हैव नो प्रॉब्लम टू कंडक्ट एनी ट्रेनिंग.
हाँ, वो वहाँ पे हमने ऑफलाइन ट्रेनिंग की है सर बहुत सारा.
ठीक है डॉक्टर रोमन. Thank you,
Thank you so much, sir. Thank you so much, sir.
Okay, my dear. Okay, thank you. Should I leave now?
Yes, sir. Thank you so much.